All right, I think it's time to start. So hi, everyone. Welcome to Creative Biolabs live webinar series. And to start, I would like to briefly introduce Creative Biolabs to all our attendees today. So Creative Biolabs is committed to supporting life science research clients worldwide through our extensive global network. We are pioneers in providing a comprehensive range of advanced technologies and products in the neuroscience research field. Our offerings include high-quality antibodies, but also proteins, cell lines, cell culture tools, and modulators. Additionally, we have customized services which can address every facet of neuroscience research to meet your scientific needs. With our integrated global resources, we aim to offer you a cutting-edge platform that can significantly accelerate your neuroscience research. Now, today's webinar topic is regulation of CNS inflammation by astrocytes. Astrocytes are critical regulators of central nervous system inflammation, capable of both promoting and resolving inflammatory responses. Upon injury or disease, they become reactive and can release pro-inflammatory cytokines, chemokines, and reactive oxygen species, thereby driving neuroinflammation. However, they also have protective roles. For example, they can secrete anti-inflammatory factors like IL-10 and TGF-beta. They can also maintain the blood-brain barrier, and they can prevent excitotoxicity. So they have a dual role in both exacerbating and mitigating inflammation, which makes them vital targets for therapeutic strategies aimed at treating neuroinflammatory disorders. Now, in this webinar today, we are very honored to have an expert speaker, Dr. Francisco Quintana, who will guide us through the intricate ways that astrocytes regulate CNS inflammation. So I will briefly introduce Dr. Quintana. So he is a professor of neurology at the Ann Romney Center for Neurologic Diseases at Brigham and Women's Hospital at Harvard Medical School, and he's also an associate member at the Broad Institute of Harvard and MIT. In addition, he also serves as the president of the International Society of Neuroimmunology. His research focuses on neuroimmunology, particularly the signaling pathways that regulate inflammation and neurodegeneration, thereby aiming to discover new therapeutic targets and biomarkers for related disorders. Dr. Quintana has been very productive. He has authored over 200 peer-reviewed articles in this field and has identified significant roles for the transcription factor AHR in inflammation control. His work has also explored how various factors influence central nervous system resident cells in health and disease, thereby leading to multiple patents and the foundation of 4 companies. In addition, Dr. Quintana has received numerous awards, including the Lady Anne Chain Prize and the Kukro-Weiner Distinguished Professor of Neuroimmunology title in 2021. Now, before Dr. Quintana begins his presentation, I want to quickly remind our audience that we will have a Q&A section at the end of the presentation. And if you have any questions during the presentation, please type your questions into the Q&A panel, and then Dr. Quintana will get to them after his talk is finished. So now let's welcome Dr. Francisco Quintana, and thank you, Dr. Quintana, for taking the time to be with us here today and for presenting your important findings to us. And yeah, feel free to start.
Thank you so much for the nice introduction. Thanks for having me here today. It's actually an honor to be in front of this audience. So what I'm going to do is actually to discuss some new findings in terms of the role of astrocytes in regulation of CNS inflammation. When we think about the CNS, obviously, and CNS resident cells, obviously we think about neurons. But then when we think about glial cells, right, we think about oligodendrocytes, abundant cells which are involved in myelination, remyelination, metabolic support of neurons. We think about microglia, which are the CNS resident immune cells. These are the brain macrophages together with some other myeloid cells. And obviously, they are going to be very important in controlling inflammation in the CNS. Our focus has been actually on astrocytes, the most abundant glial cell of the CNS. Astrocytes tile the CNS, and actually they participate in multiple homeostatic functions. They actually provide nutrients and metabolites that support neurons. They're actually very important for the maturation and the stability of the blood-brain barrier, and also for the maturation and function of synapses. Now, part of our work has been focused on demonstrating that actually astrocytes are not an homogeneous population. There are actually multiple subsets of astrocytes. Indeed, we have identified astrocytes that can either promote or suppress inflammation in the CNS. And they do so through multiple mechanisms. They can actually promote inflammation via the activation of CNS resident immune cells, such as microglia. They can also recruit peripheral immune cells into the CNS. And on top of that, astrocytes can also have their own intrinsic pro-inflammatory responses. We also identified astrocyte populations that can actually suppress CNS inflammation, and they do so again via multiple mechanisms associated with the production of cytokines and other molecules, or even the direct induction of T-cell apoptosis. Whether these pro and anti-inflammatory functions are beneficial or pathogenic, it really depends on the disease or in the disease context. For example, anti-inflammatory astrocyte functions could be beneficial in the context of autoimmune diseases such as multiple sclerosis, yet they could promote CNS pathology if we're thinking about glioblastoma, favoring, right, helping tumors evade the immune response. So as part of their function and regulation, actually, astrocytes do not only regulate CNS inflammation, they actually respond to inflammatory or to immune signals provided by CNS resident and CNS recruited immune cells. Obviously, the obvious partner of astrocytes in terms of the regulation of CNS inflammation is microglia. And we and others have identified multiple factors which are produced by microglia in order to regulate astrocyte function. On top of that, astrocytes also are regulated by factors produced by immune cells, peripheral immune cells recruited to the CNS, such as T cells, monocytes, and NK cells. Hence, the idea I want you to keep is a concept that astrocytes actually establish a bidirectional interaction with the immune system. And those interactions involve CNS resident and CNS recruited immune cells. Those interactions also have important consequences, not only for your classic autoimmune disease of the CNS, which is multiple sclerosis, but also for other diseases such as neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, for tumors targeting the CNS, such as the main primary brain tumors, such as glioblastoma. And on top of that, those interactions are going to be central for our ability to fight off viral infections in the CNS. So with that in context, with the idea that astrocytes establish bidirectional interactions with the immune system, what I would like to discuss with you today is basically 2 points. First of all, how can we identify astrocyte interactions with the immune system? How can we identify mechanisms by which interactions between astrocytes and immune cells can actually promote or suppress astrocyte pro-inflammatory responses? And the second question I would try to discuss with you today is whether astrocytes remember previous interactions with the immune system. Is there any kind of memory that astrocytes acquire after interacting with the immune system that will change and modify the responses in the future? So to get started, right, to ask the question of how can we study astrocyte responses, control or the control of astrocyte responses by immune cell interactions. To get started there, we have to start by like actually envisioning the challenge, right? We can work with astrocytes in vitro. We can co-culture astrocytes with immune cells in vitro, but that's unlikely to recapitulate, first of all, the heterogeneity of astrocytes I mentioned, and that has to do with the fact that most times in vitro astrocyte cultures are established either from neonates or from very well-defined adults. But then the other point is that when we co-culture astrocytes with cells in vitro, obviously there are so many cells we can co-culture them with, and we tend to lose the 3D structure, right, that is so central to the organization of the CNS. So the question then we've tried to address a few years back is how can we study the complex interactions, the complex cell-cell interactions that regulate astrocyte responses or the responses of many other cells in the CNS? To address that challenge, we developed a technique which we call rabbit-seq, which basically takes a rabies virus and modifies it so it can basically pass a molecular barcode among cells that are interacting in the CNS. And just to tell you a little bit how we generated this virus, basically we took the classic rabies virus And then we engineered it in multiple ways. First of all, we engineered its envelope. So now it can target astrocytes, which express a specific envelope-specific protein, which is called TBA. On top of that, we actually deleted the protein G of the virus, so it can only replicate in cells that on top of expressing the receptor for the pseudotype virus, they also express protein G. And finally, what we did is we actually incorporated an M-cherry protein, so we can actually isolate cells that are infected. But at the end of that M-cherry protein, we incorporated a genetic barcode. And that's important because instead of having one single rabies virus, we're going to have, or we work with a library of 1 million different rabies viruses, each one characterized by a specific genetic barcode. Hence, we can actually isolate those cells that are red, they express M-cherry because they've been infected, and then we can look for barcode sharing in order to identify cells that were interacting in vivo. So let me show you how this works in vivo with an example. And in this case, I'm going to show you an example of the EIE model of multiple sclerosis. This is a model where on day 0, you immunize mice with a myelin peptide, and about 10 days after that, you start to see the accumulation of neurologic disability. And this is actually a model that recapitulates many aspects of multiple sclerosis. Obviously, not all aspects, but many of the drugs that we have nowadays working on multiple sclerosis are actually coming out from this model. So we analyzed by rabbit-seq these the astrocyte interactions in this model. And the first outcome, right, the first piece of data you get is something you've seen thousands of times by now. It's a UMAP, it's a Disney plot where each dot represents a cell for which you have transcriptional data. However, what makes rabbit-seq different is that each one of those dots, each one of those cells is annotated with a unique genetic barcode. So we can actually reconstruct the interactions that each one of those dots were establishing in vivo. Hence, we can turn a UMAP or a Disney plot like this into an interaction map like this one, where we're still preserving single-cell RNA-seq data. We're still preserving the genetic information, the transcriptional information about each one of the cells interacting in vivo. And that's important because then we can actually say, we can first define a specific subset of cells, and then we can ask the question of what are the interactions that that specific cell subset was establishing in vivo. And indeed, that's the first question we addressed with this system. We actually decided, first of all, to look into astrocytes, and then to focus on astrocytes that, because of their transcriptional signature, displayed a very high pro-inflammatory signature. So this is the most pro-inflammatory subsets of astrocytes we could identify based on the transcriptional signature. And then we decided to ask the question, right? Based on this population, what are the microglial cells that interact with it? Is there anything particular about the interactions between this highly pro-inflammatory astrocyte population and microglia that will actually explain the development of this highly pro-inflammatory and disease-promoting astrocyte subset? So we ran our analysis, and to our surprise, on the top of our list, we didn't detect cytokines, right, as mediators of interactions with microglia promoting these very pro-inflammatory astrocyte subsets. Actually, what we detected was a reactivation or a co-opting of a family of signals, which are called axon guidance proteins, which are obviously, as the name indicates, very important for CNS development. Yet, in the context of immunology, axon guidance proteins have been described to play important roles in the regulation of the immune response. So hence, this is probably an example of a protein or a family that, because of its context, it was given a certain name, but in biology, it plays many other functions. In particular, our studies identify within axon guidance pathways, identify this specific family involving the F receptor and its ligand ephrin. To be more specific, our prediction at this time identified the receptor called FB3 as a receptor expressed by astrocytes, which could mediate interactions with microglia via a ligand called Ephrin-B3. And this is very interesting for several reasons. First of all, FB3 is actually a tyrosine kinase. So that means that here in the intracellular domain, there's a kinase domain that is very important for the signal transduction of the activity mediated by this receptor. And obviously, tyrosine kinase and kinases in general are actually interesting targets for the development of drugs and therapeutic interventions. So that caught our attention. The second point I want to highlight is that the ligand interacting with this specific receptor, Ephrin-B3, is actually a membrane-bound ligand, and is actually a membrane-bound ligand that has an intracellular domain that can also mediate signal transduction. So interactions mediated by Ephrin-B3 and FB3 are actually bidirectional interactions. They can trigger signaling and modify the responses of both cell types participating. In this specific case, they can modify the responses of both astrocytes and microglia. So with that background in mind, so then at the time that was a prediction from our model, let's see whether we could validate the functional role for this specific signaling pathway. So the first thing we did was actually to analyze tissue samples from MS patients. And to cut it short, what we found was that if we looked at the CNS of MS patients, we detected a significant upregulation of FB3, the receptor in astrocytes localized in multiple sclerosis lesions. And we also detected the upregulation of Ephrin-B3, its ligand, expressed in microglia also located in those lesions. So both the receptor and the ligand seem to be upregulated in the lesions of MS patients. So now to perform some more mechanistic studies, we went back to the EIE model of multiple sclerosis. And what we found is that if we inactivate either the receptor in astrocytes or the ligand in microglia, that results in a significant amelioration of the disease and a significant decrease in pro-inflammatory astrocyte responses that are known to promote disease pathology. These and other studies that we performed, also using in vitro systems, right, in astrocytes and microglia, even using human in vitro systems, led us to conclude that interactions mediated via FB3 in astrocytes and Ephrin-B3 in microglia can actually promote pro-inflammatory responses in both. And indeed, they seem to be an exciting candidate therapeutic target. We were actually able to develop the small molecule, which at the time we call A38, which is a CNS penetrant specific inhibitor of FB3. This small molecule ameliorated inflammation and pathology in multiple neurologic diseases, and is actually currently being further developed and optimized with the idea of putting it into patients. Now, if we step back, right, what I want you to live with, the concept I want you to live with is the idea that we developed this technique, which we call rabbit-seq, which enables us to interrogate cell-cell interactions in vivo within the complex microenvironment of the CNS. Now, you might argue, well, you might not reach all the interactions involved because, for example, you're not looking at the right time point, you're not looking in the right model, you're not looking into the right location. So obviously there are many more interactions that might occur and that do indeed occur between astrocytes and microglia that you might not be detecting with this system. In order to address that question, in order to address that challenge, we developed a new technique which we call SpeakSeq, which basically enables us to perform genome-wide forward genetic screens of mechanisms of interaction between our favorite cell types. In this very specific case, we used it again to interrogate the interactions, or actually to define the interactome, all potential mechanisms by which a microglia could control astrocyte responses. And the way it works is the following. We basically co-culture cells in droplets. So each droplet works as a cell. And then obviously we have millions of cells, millions of droplets, millions of pairs of astrocyte microglia pairs, where with a particularity, right? With a particular thing that actually in each one of these droplets, we have inactivated one single gene in the microglia. And then the astrocyte co-cultured with it harbors a reporter that will tell us how the inactivation of one gene in microglia affects astrocyte responses in those astrocytes co-culture in those droplets. So we can perform, and we did indeed perform that in a genome-wide manner. So we can basically ask the question of within the genome, what are the genes that when perturbed in microglia can affect astrocyte responses? And obviously you could argue that there will be a certain bias in these studies because there might be interactions that we do not detect because they occur only in vivo and not in this artificial setup. We do accept that, but I think that that's one of the best tools we have in order to get candidates of mechanisms of interaction that then we can go and validate in vivo. So let's follow up in terms of how can we validate those interactions in vivo. So the first thing we did was, as I mentioned, to run this genome-wide screen. In order to run it also, we have to develop the droplet cytometer that would allow us to, first of all, detect droplets that have only one microglia and one astrocyte. And then on top of that, to detect the activation of the specific reporter in the astrocyte. So by using this system, we established for the first time the whole interactome, the whole set of potential genes which, when expressed in microglia, they can affect the ability of microglia to interact with astrocytes. And then we decided to focus, first of all, on those genes coding for secreted molecules. We wanted to focus on secreted products of microglia that act on astrocytes. And on top of that, we decided to focus on molecules that actually are secreted by microglia with the idea of trying to suppress astrocyte pro-inflammatory responses. We wanted to look for regulators built in the CNS in order to try to limit astrocyte pro-inflammatory responses. That led us to come up with a list of 4 candidates which are produced by microglia, each one of them acting through a specific receptor in astrocytes. So the question is then, how can we know which one of these candidates, right, is the one most relevant for the regulation of astrocyte responses by microglia? To address that question, what we decided to do was to develop the first in vivo perturb-seq study in astrocytes, where we basically inactivated each one of these ligands and receptors. We use a specific barcode to label each one of the guide RNAs associated with the inactivation of each one of these genes. And then we analyzed by single-cell RNA-seq which one of these genes would perturb resulted in the largest deregulation of astrocyte responses. And what we found was that actually this specific product, which is a protein called amphiregulin, when acting via a receptor called the EGFR receptor, turned out to be a candidate regulator of astrocyte pro-inflammatory responses. Obviously, the other ones, they also had some effects, but this one was the one that scored the highest in our studies. So now a little bit of background. Amphiregulin is actually a protein that is well known to immunologists. Actually, for example, in the gut, it has been known to play important roles in tissue repair after gut inflammation. In the context of the CNS, regulatory T cells were reported to actually produce amphiregulin in order to deactivate astrocytes. Yet, to cut a long story short, we showed by generated conditional knockout mice that this mechanism does not seem to operate in the context of multiple sclerosis and EAE. So this is important for stroke, but not for multiple sclerosis. And the last piece of information I want you to remember before we move forward is the fact that actually it was reported that IL-33, a cytokine, was known to be an inducer of amphiregulin. And what is interesting to know about IL-33 is that this is a cytokine that is kept inside the cell by cells, but when the cells are stressed, let's say, for example, in the context of inflammation, tissue damage, IL-33 is released and can trigger multiple responses. So with that background in mind, let's go back and try to understand or to validate this finding of amphiregulin produced by microglia acting as a negative regulator of astrocyte pro-inflammatory responses. The first experiment we did was actually to inactivate amphiregulin in microglia. And as you can see, when we do so, there's a significant worsening of EIE. And that results in a significant deregulation of astrocyte pro-inflammatory responses. These and additional complementary experiments performed in vitro, both with mouse and human astrocytes, suggested that amphiregulin operates as a negative regulator produced by microglia in order to suppress astrocyte pro-inflammatory responses. Now the question is, how does microglia know that they have to release amphiregulin? How do they know that astrocytes are becoming activated, that they are becoming pro-inflammatory, and that has to be somehow dampened? So to address that question, we went back to our rabbit-seq dataset, right? And basically, we searched for it, and first of all, we identified microglia producing amphiregulin connected to astrocytes, and then we looked at the upstream regulators of those amphiregulin producing microglia. And we identified as a candidate regulator IL-33, which, as I mentioned before, has been previously described to be an inducer of amphiregulin production. So that led us to question whether astrocytes in the context of inflammation secrete IL-33 as a way of inducing microglial production of amphiregulin, some kind of negative feedback loop in order to try to suppress astrocyte pro-inflammatory responses. So to validate that cellular circuit or that hypothesis, the first experiment we performed was actually to generate a conditional knockout mouse in where we inactivated the IL-33 receptor specifically in microglia. And as you can see, when we do that, that results in a significant deregulation of EIE, a significant worsening of EIE, a significant worsening of astrocyte pro-inflammatory responses. Indeed, if we treat microglia in vitro with IL-33, we can actually induce amphiregulin production. Now, to further validate this cell circuit, what we did was actually to knock out IL-33 specifically in astrocytes in order to define astrocytes as a relevant source of IL-33. And again, we got a very similar phenotype. When we inactivate IL-33 specifically in astrocytes, there's a significant worsening of EIE, and that results in a significant deregulation of inflammation. So these and another experiments led us to conclude that there is a cell circuit by which astrocytes, in the context of inflammatory responses, they secrete IL-33, which then acts in microglia in order to trigger amphiregulin production as a way of trying to dampen those pro-inflammatory responses. Now, if we step back a bit, what we found is that actually we established this new system, which we call SPIC-SIG, which enables us to interrogate the mechanisms that mediate cell-cell interactions between our favorite cell pairs. And as you can imagine, we're using it to define novel mechanisms by which dendritic cells control T-cell responses, by which tumor cells control tumor-specific immunity, and so on and so forth. So we believe that this is a very powerful and exciting platform. So with that, we discussed, I think we had two nice examples using, focusing on CNS resident immune cells, such as microglia, right? But these are two nice examples of how immune cells can control astrocyte responses. Now, what I would like to discuss is whether actually astrocytes have some kind of memory, right? They keep memories of their interactions with either CNS resident cells, CNS resident immune cells, such as microglia, or peripheral immune cells recruited to the CNS. And that would be important because that would lead us to understand how one or a few encounters, right, with immune cells in the context of infections or trauma or even other contexts, can actually have long-term implications for the control of astrocyte inflammatory and neurotoxic activities. So this idea of whether astrocytes remember previous interactions with the immune system bring us back to a very basic concept in the context of immunology, which is the idea of immune memory, right? Immune memory, the most common form we are familiar with is what we call adaptive immunity, basically memory driven by T cells and B cells, which is what we activate or what we exploit when we get vaccinated. Basically, we're vaccinated or get an infection, antigen-specific and bug-specific T cells and B cells are expanded. Some of them are long-lived, and those long-lived cells are going to afford us protection against future encounters with the same bug. Now, more recently, it was rediscovered. I mean, that was a very old finding that was kind of like rediscovered more recently, that even innate immune cells, such as microglia or monocytes, they can also have a form of immune memory, basically meaning that following a first immune stimulation, they can go back to a resting state, but then upon restimulation, they can display faster, stronger, broader immune responses that actually help you fight inflammation. Now, the question we want to address or discuss here today is whether astrocytes, which are non-immune cells, whether they could still develop some form of immune memory. So in order to address that question, the first thing we did was to develop a new experimental model. And this is a model where we injected into the CNS I1 beta and TNF. And we decided to use these two cytokines because we have previously shown that when you activate astrocytes in vitro with I1 beta and TNF, you induce transcriptional responses that are pretty much close, or they're pretty close to the ones we detect when we analyze astrocytes from multiple sclerosis patients. So our experiment was to inject I1 beta and TNF either once and then isolate astrocytes the day after, or to actually inject it once, wait for a week, so all those astrocyte responses go down, and then re-challenge those mice, and then reanalyze them a day afterwards. And we repeated these also 3 times, so on and so forth. And also we played with lengthening the period between the first and the second stimulation. And what we found was very interesting. Obviously, if we stimulate them once, there's a significant upregulation of pro-inflammatory responses that we and others have previously characterized. And we can detect them by ATAC-seq, by RNA-seq. The interesting thing, as you start to repeat that stimulation, we detect 3 types of responses. First of all, there's a significant, a very marked downregulation of multiple genes, many of which are associated with homeostatic astrocyte functions. They're associated with the ability of astrocytes, for example, to support neuron metabolic needs, to produce neurotrophic factors, so on and so forth. On top of that, we also see that genes that were initially induced in the context of stimulation are actually induced at a much higher level, right? And even if we were to do kinetics, those things happen even faster. And the last thing we see is that there's a group of genes that potentially were not initially expressed following the first stimulation, but now start to be expressed. So this starts to look a little bit, right, like those memory responses that were previously described in innate immune cells. Yet, remember that astrocytes are not immune cells. This is an exciting finding. This is an exciting model, yet there are many limitations I could describe about, I could associate to this experimental system. And the first one I would give you is that it could certainly be that when we're injecting I1 beta and TNF, we're actually activating and educating the microglia. And the changes in astrocyte responses we see are simply a reflection of changes in microglial responses. So the question or the challenge is, how can we interrogate astrocyte intrinsic memory phenotypes? To address that question, we developed a new system where we basically used cells or astrocytes derived from NF-kappa B reporter mice. So these are mice that when NF-kappa B signaling is activated, will upregulate the expression of GFP. And that's important because I1 beta and TNF are strong inducers of NF-kappa B. They're strong activators of NF-kappa B. So this system then enables us to basically activate astrocytes 1, wait for those NF-kappa B activating cells to become green. We can sort them out. We can rest them so they become non-green again, so they do not express any other gene. And then we can restimulate them and see whether somehow these genes, these astrocytes, which for sure we know, we know there was a previous immune activation, we can see whether the future responses have been altered. And the answer is yes. If we look at those astrocytes, those actually display a significant upregulation of their pro-inflammatory responses, and those involved cytokines such as IL-6, which are involved in the activation of other cells such as microglia within the CNS, cytokines such as chemokines such as CCL2, which are very important to recruit pro-inflammatory cells from the periphery into the CNS. And we even detected changes in NOS2, which is an enzyme which is involved, for example, in pro-inflammatory and neurotoxic activities of astrocytes. So this is starting to look a little bit like this memory phenotype that was described in immune cells. Then, in order to understand how this is regulated, we performed ataxic experiments, and interestingly enough, we detected a significant change in chromatin accessibility associated with not only effector molecules, right, like cytokines, but also with receptors of immune signals. So this suggests that actually not only, and this goes along with the idea that not only these astrocytes can develop differential or different responses upon restimulation, they're also going to be more sensitive to these immune stimuli. Hence, we are talking about long-term changes, and these actually changes in chromatin accessibility started to look or suggest epigenetic reprogramming. Now, we performed then a bioinformatic analysis to identify candidate regulators of this phenomenon. We identified NF-kappa B. That should surprise no one because, as you know, NF-kappa B is actually strongly activated by I1 beta and TNF. But what we also detected was an enzyme which is a histone acetyl transferase, which is called P300. And basically, this enzyme catalyzes the addition of acetyl group to histones. And by doing so, then basically it opens the chromatin and makes it more accessible for expression of the genes encoded within that chromatin. So we then interrogated whether P300 is indeed regulated or involved in the regulation of this memory, or this astrocyte memory, as we started to call it. And to do so, we performed multiple experiments. One of those was actually to inactivate P300 in astrocytes. That results in a significant amelioration of EIE. But what is more important, that results in a significant reduction of genome-wide histone acetylation. This is telling us that in these conditions, P300 actually is a dominant driver of histone acetylation in astrocytes. And most importantly, that results in a significant decrease in a transcriptional signature that we have associated, or we have detected associated with these memory astrocytes. So taken together, what these data suggests is that in the context of inflammation, astrocytes acquire epigenetic modifications, which are actually driven, one of the mechanisms involved, and we're going to discuss additional mechanisms later. One of the mechanisms involved is actually histone acetylation catalyzed by an enzyme called P300. This histone acetylation actually is going to allow or enable astrocytes to display stronger, faster, broader pro-inflammatory responses upon restimulation. And those are actually going to amplify pro-inflammatory and neurotoxic astrocyte activities. At the same time, these very same modifications are actually going to interfere with astrocyte homeostatic functions associated with the metabolic support of neurons and the production of neurotrophic factors. Now, if we look a little bit deeper into this mechanism, right? I mentioned that P300 is an enzyme that catalyzes the addition of acetyl groups into histones. P300 usually uses two main sources of acetyl-CoA in order to acetylate histones. It uses either acetyl-CoA provided by an enzyme called ACSS2, which basically brings acetate and metabolizes acetate into acetyl-CoA. And the second source of acetyl-CoA, the second dominant source of acetyl-CoA used, incorporated into histone acetylation, is actually an enzyme called LCLY, which is going to link cellular metabolism, and in particular mitochondrial metabolism, with histone acetylation. And it does so because it takes citrate produced by the TCA cycle, and actually it uses it to generate acetyl-CoA. So in order to interrogate which one of these two enzymes was the dominant one providing acetyl-CoA groups in order to control astrocyte memory, we basically CRISPR inactivated both of them, induced EIE, and basically analyzed their phenotypes. And to cut a long story short, we found that LCLY inactivation has the strongest effect in this memory phenotype we identified. If we analyze both disease development, but most importantly, if we quantify this memory establishment using our transcriptional signatures. So taking together what these findings tell us is that actually there's this coordinated activity, right, of LCLY, which is going to provide acetyl-CoA for P300 to acetylate histones and then to control this epigenetic program. That's interesting because we can then actually even stain for LCLY and P300 and then identify and quantify these memory astrocytes, as you can see here in the context of EIE. But also we can use this very same staining to quantify them in the context of human neurologic diseases. The challenge, though, is that LCLY and P300, although they're very useful for staining, they're actually intracellular proteins, so you cannot use them in order to sort this subset of astrocytes and really characterize them in depth. To address that challenge, last year we developed a new technique, which we call FIND-seq. And basically, it combines nucleic acid cytometry with microfluidics in order to enable you to isolate any cell of interest based on the expression of marker RNAs that you know are specific to that cell population of interest. Obviously, that's a tool that is useful to many other projects beyond this one, because we all usually tend to identify populations of interest when we perform single-cell RNA-seq studies, which many times we cannot isolate based on the expression of specific surface markers. So beyond these studies I'm going to talk about now, this is a tool that is very interesting on its own. So now, how can we use this tool to analyze in depth these memory astrocytes? We basically decided to use this tool to analyze and isolate cells astrocytes characterized by the expression of LCLY and P300. And when you perform these studies, right, this is literally the first piece of data you get. You get a plot where you can see the expression of LCLY and P300. However, what I want you to notice is that this is not a flow cytometry plot. Here you're not looking at the detection of proteins based on the use of specific antibodies. On each one of these axes, what you see is the expression of specific RNAs detected with the digital qPCR. So here you see the expression of the RNA encoding, the mRNA encoding LCLY, and here the one encoding P300. So we sorted these different populations, including the population that is a double positive, and then we use it to get an in-depth transcriptional analysis of the genes they expressed. And that allows for the first time to have a very detailed and in-depth signature associated with this astrocyte memory. That's important because we could identify additional regulators of this epigenetic program. We identified indeed additional readers, additional erasers that are involved not only in writing and enforcing this epigenetic program, but even in deleting it, which obviously is going to be important in order to regulate overall the number and strength of this astrocyte memory. Now, the second consequence of these studies is that now we have a transcriptional signature we can use in order to identify this population in other data sets. So the first thing we did was actually to go to our to a previous data set we generated in the context of EIE, this experimental model of multiple sclerosis. And as you can see, we identified a specific population that bears the signature of astrocyte memory and seems to be significantly expanded in the context of EIE. Now, we can perform the same type of analysis, and actually we can go and analyze clinical samples. And then in the context of multiple sclerosis, again, we can identify a population that bears a signature indicating that this is actually a memory subset in the context of MS. And what is very important or interesting is that when we analyze where it is located, this is located in chronic active lesions. These are lesions which are supposed to be very important for driving disease progression, for driving neurodegeneration in multiple sclerosis, and this is where astrocytes are known to play important roles. So this is an important finding because it suggests an important role of astrocytes or memory astrocytes in MS pathology. So these and additional studies have led us to identify this astrocyte memory. We like to call it astrocyte immune memory to differentiate it from roles of astrocytes in cognitive memory, which obviously are going to be very important. And we now know that similar mechanisms of memory operate in the context of neurodegeneration and cancer. Obviously, one of the first things we want to do now is to actually define cell-intrinsic mechanisms that regulate it. And one of the first experiments we performed in order to address it was actually to perform a genome-wide CRISPR screen of candidate regulators of astrocyte responses. That led us to identify, among multiple candidates, the one that caught our attention was our strongest hit, because this protein, CLEC16A, is actually a protein that is well known for those working on multiple sclerosis. And why is that? Because in multiple genome-wide screens of genes associated with multiple sclerosis, CLEC16A is one of the top-ranking genes. Indeed, is one of the top genes when you exclude HLA, which is basically associated with irregulated T-cell responses. So obviously, we were very interested, right? Because that suggests that genetic polymorphisms associated to multiple sclerosis could potentially regulate an astrocyte memory phenotype. So we actually performed a series of mechanistic studies in order to define how is it that CLEC16A controls astrocyte memory, and I'm actually going to summarize all of them in this summary slide. And to cut it short, basically what we found is that what CLEC16A does is it actually operates on the mitochondrial side of this astrocyte memory. Basically, it promotes the removal of all mitochondria, of dysfunctional mitochondria, and by doing so, it limits the generation of pro-inflammatory responses, it limits the generation of astrocyte memory. And just to give you one slide of how that happens, if we generate a conditional knockout mouse in which we inactivate CLEC16A specifically in astrocytes, as you can see here, there's a significant worsening of EIE. But what is more important in the context of this talk, we get actually twice as many astrocytes displaying a memory phenotype. So CLEC16A is probably one of many negative regulators that we have identified operating to limit the generation of astrocyte memory. As I mentioned, we have identified at least one eraser, one epigenetic eraser that is involved in deleting or kind of trying to remove the epigenetic program associated with astrocyte memory, and that's something that we're mechanistically interrogating as we speak. And now to finalize, obviously, we worked here with cell-intrinsic, or I wanted to discuss with you cell-intrinsic mechanisms, right, that regulate astrocyte immune memory. But in the context of an inflamed tissue like the CNS, it's likely that cell-cell interactions are also going to contribute in order to establish this phenotype. So we're using rabbit-seq and pig-seq, which we discussed earlier on during my talk today, as a way of defining cell-cell interactions that boost and stabilize astrocyte memory. And the last thing we are doing as part of our studies in this subject is actually to study how environmental factors perturb astrocyte memory. And the reason for that is because we were the first to describe that the gut flora can actually secrete metabolites that act on the CNS in order to regulate astrocyte responses. So hence, we believe that metabolites or even environmental chemicals can get to the CNS and affect the formation of memory. And that's very important when we think about specific microbiome perturbations or exposure to environmental pollutants that have been associated with neurologic diseases. This could be a mechanism that contributes. But on top of that, we've been for quite some time, we've been interested on the idea of designing probiotics as a way of providing specific metabolites in order to modulate inflammation. As I mentioned, these epigenetic programs, right, are closely linked with the metabolism of astrocytes and actually are closely regulated by it. So we're working with engineered probiotics designed with the goal of trying to control this memory phenotype. And we believe they might be a very useful tool to do so by directly targeting the gut-brain axis. And the last point I want to make is that this is not a phenotype that is only specific to astrocytes, right? I believe this is an important feature that is probably relevant to other cells in the CNS and also to other cells in the periphery. So we might start thinking about inflammation as actually not a property of immune cells acting on specific tissues, but actually of the whole tissue becoming inflamed, the whole tissue carrying a scar of previous inflammatory events. And hence we have to identify and develop strategies to really try to remove those tissues if we want to remove those scars of previous inflammatory events, if we want to come up with better therapies for chronic conditions, such as the neurologic conditions where astrocytes play very important roles. And with that, I'm going to close my talk. We just to summarize, we discussed, first of all, how interactions with the immune system, today we focus mostly on microglia, how they can actually promote or control astrocyte responses. And then more in the last part of my talk, we discussed how those interactions with immune cells can leave some epigenetic scars that change or memories that actually change the responses or the future responses of astrocytes to REST stimulation. And with that, I want to finish by acknowledging the scientists in the lab that ran those studies. Hong Lee was the one running the studies on astrocyte memory, and Atsushi Kadowaki was the one running the studies on CLEC16A. And with that, I'll be happy to take any questions you might have.
Thank you, Dr. Quintana. That was a very insightful presentation. I hope our audience found it as engaging as I did. With that, we are open for questions. So for our audience, if you have any questions, feel free to type them in the Q&A. And then actually to start with, I do already have a couple of questions because it was very interesting. And you already mentioned, you already explained a couple of things in your summary, especially regarding the fact that other cells besides besides the astrocytes in the brain can actually contribute to immune memory, such as maybe oligodendrocytes or neurons. I'm actually curious to hear your thoughts on how these how you would envision that works.
So how do I envision these networks?
Yeah.
Yeah, so I think that's a very interesting question because we see those networks as not only disease specific, but also even microenvironment specific. So for example, just to give you an idea, we do identify different networks, for example, when we focus on chronic active lesions, as opposed to other areas of the CNS in MS. And we do know that those networks are different and actually involve different mediators when we compare AD, Alzheimer's disease, with multiple sclerosis or Parkinson's disease. So that's why I think it's unique to have, we're very fortunate to have some of these tools I mentioned, because we can identify specific mediators that are disease and location specific. And then we can actually go back and look into the specificity of the memory responses themselves, because obviously there are elements that are shared across diseases, but some inducers are going to be disease specific, right? And also some of the effector responses associated with those memories are going to be disease specific. So that's a little bit of an insight of where we're going into now.
Yeah, indeed. Okay, yeah, that is already answering several other questions. Because I was assuming because you're using the EAE model or the IL-1 beta and TNF stimulation, so the responses to that would be depending on the stimulus, like it's depending on the IL beta and the TNF. But if you use a different stimulus, you would also respect other factors to be involved. So it would not just be P300, but potentially other epigenetic regulators, right?
Exactly, and indeed, we have identified already this phenomenon and these memory astrocytes, for example, in the context of Alzheimer's disease and in the context of Parkinson's disease. And some things seem to be shared, but some of the drivers seem to be really disease specific. And that makes it very exciting, I think.
Yeah, that's very nice. Yes. Yeah, then another question. Do you see for the Ephrin-B part, so you have the Ephrin-B and the Ephrin-B3 expression, do you actually see any regional differences for this for these proteins? Is it limited, for example, to white matter, or is it also present in gray matter or in certain brain regions or spinal cord? Yeah.
That's an excellent question. So indeed, we do, something we notice is that we detect these interactions mostly in lesions. In the normally appearing white matter, we do not detect them. We actually identified semaphorin 4D interacting with plexin B1 and B2 as something which is more abundant in the normally appearing white matter. So that again brings the idea that different cell interactions are established in different environments, right? And then that's important for us when we start to think what to target and how to target it.
Yes, and in relation to that, do you know of any compounds that exist that can block this interaction between F3
yes.
and...
So we generated, I briefly touched upon that. We generated, first, as part of our studies, we performed a small molecule screen with a library of proprietary compounds we had. And we identified one compound, which at the time we called A38, which is brain penetrant, and it's very strong inhibitor of FB3 tyrosine kinase domain. We have actually performed multiple rounds of medchem with the goal of actually bringing that into a clinical trial.
Yeah, okay, very nice. Yeah, maybe one last question before we finish up. Do you have any idea if IL-33 or the other protein AREG has been found to be changed in patients for multiple sclerosis or in other neurodegenerative conditions, or do you know of any mutations that exist that would decrease their function?
That's a very good point. Not that they had been changed, like not intrinsic mutations, but for example, there's additional data about IL-33 in the context of CNS inflammation and neurodegeneration, and in the context of GBM as well, glioblastoma.
Yeah, yes, yeah. All right, okay. Thank you so much. And with that, I think our time is up. So thank you very much for this wonderful talk. It was very interesting. We've learned a lot from that. And then I would like to mention that, so we do actually regularly invite experts in life sciences and biotechnology to share their latest findings in our live webinar series. And we encourage everyone to visit our website and subscribe to our social media channels so you won't miss any upcoming events So that's it for today. And I would like to thank everyone for joining us. And again, Dr. Quintana for this amazingly interesting talk. And then, yeah, we will see you next time. Thank you all.
Thank you. Bye-bye.
Bye.